New week, new media storm around a scientific study. According to all the headlines, a new study has implicated seed oils in cancer. First rule of Fight Club, guys, you gotta take the media headlines with a massive grain of salt. Always double check, always look into it. Don't just repeat what they say, because this stuff is all over the place. This study in question didn't look at seed oils, didn't mention seed oils once in the entire study. Didn't look at any other foods either. It's not a diet study at all. Okay, so what did they do? They took cells from people who had colorectal cancer. So this is cancer of the large bowel. And so they took a sample of the cancer cells, of the tumor cells, and also a sample of normal cells of the same individuals off to the side in the large intestine. And they compared the types of lipids, the types of fats that exist inside those two types of cells. So this technique, this approach is called Lipidomics. It's basically getting a huge laundry list of all the different types of classes of fats inside these two different types of cells. So they found hundreds of different types of lipids that were either higher or lower inside the tumor cell compared to the normal cell, including a number of phospholipids, which are types of fats that exist, for example, in the cell membrane. Some of these phospholipids contain saturated fats, some contain monounsaturated fats, some contain omega-3s, and some contain omega-6s, like linoleic acid, for example. And linoleic acid is a fat that is pretty high in most seed oils, so this is where they're getting this loose connection. Linoleic acid is an essential fat, so our body doesn't produce it. We need to get it from food. But finding out that it's higher in some phospholipid species inside one type of cell than another tells us nothing about food. We have no idea what these participants were eating, what kinds of foods, how much. None of that is ever measured. We also have no idea if each of these lipid molecules that were higher or lower inside the cancer cell are good or bad or neutral. These patients already had cancer, and this study is just a snapshot in time. There's no follow-up. It's just one measurement. So we don't know if these phospholipids caused the cancer or are slowing down the cancer or are neutral, just sitting there as a byproduct of metabolism. We have no clue. This study design can't get at cause and effect. It's not meant to. It's not designed to do that. It's supposed to characterize the lipid pools, the lipid content of those two cells to generate more knowledge and then to generate clues to then test in other types of studies. Maybe eating more linoleic acid and more seed oils causes colorectal cancer. Maybe it prevents colorectal cancer. Maybe it makes no difference. We can't tell from this type of study. There are many other misunderstandings about the study that also got fueled by the media headlines and by most of the social media commentary. In our bodies, linoleic acid is converted to arachidonic acid. So that's another type of lipid. And one simplistic idea is that arachidonic acid is bad. It's unhealthy, period. So let's just avoid linoleic acid, cut back to avoid arachidonic acid being produced. The problem is this conversion is pretty fixed. And so even with huge variations in the intake of linoleic acid, our level of arachidonic acid is pretty constant. This has been shown over and over in research. These authors explain it. The production of arachidonic acid from linoleic acid is tightly regulated. Wide variations in dietary linoleic acid do not materially alter tissue arachidonic acid content. The extent of conversion of linoleic acid to arachidonic acid is about 0.2%. In fact, our plasma levels, our blood levels of arachidonic acid, are a better reflection of our intake of animal products like meat, red meat, for example, than our intake of seed oils. That's because there's preformed arachidonic acid in animal products like meat and fish. So those foods already contain arachidonic acid. It doesn't need to be converted. And plus, this notion that arachidonic acid is unhealthy, it's this bad molecule, it's just not true. It's a huge oversimplification. Arachidonic acid can be converted into lots of different molecules. Prostaglandins, leukotrienes, thromboxanes, lipoxins. Some of these molecules are pro-inflammatory, some are anti-inflammatory. So reducing all of this complexity, all of this biology to bad is just cartoonizing science. Scientists who study this topic in depth, they write about this simplistic notion almost making fun of it, almost making light. If one takes off the blinders and examines the entire arachidonic acid metabolome, metabolome is just all of these molecules, all of these metabolites of arachidonic acid. So all of these molecules that come from arachidonic acid. One finds a constellation of metabolites, including, and then they list a bunch, prostaglandins, leukotrienes, and on and on and on. Some are pro-inflammatory, some are anti-inflammatory. To label the entire class of omega-6 fats as pro-inflammatory is painfully naive. In fact, one of the takeaways of this study that made the headlines is the authors propose that inside the cancer cells, there's a shift. There's a lipid class shift towards lipid metabolites that are more pro-inflammatory and away from lipid molecules, lipid metabolites that are more anti-inflammatory. Here's a quote from the study. Our lipidomics data from human tumors indicates a strong prevalence of pro-inflammatory mediators, and they give a couple examples. 5-HEAT, LTB4, LT stands for leukotriene. So these are examples of lipids that are more pro-inflammatory, and on the other hand, extremely low levels of LXA4, LX stands for lipoxin. So LXA4, LXB4, two examples of lipoxins. These are more anti-inflammatory and they're low in the tumor cells. But all of these molecules that they mention here, all of these metabolites are downstream of arachidonic acid and linoleic acid. They're all derived from those molecules. Here's a diagram of the arachidonic acid pathways. Don't worry, we're not going to go over all of this in detail. Just to show you real quick, here's 5-HEAT that is mentioned in that passage. Here's LTB4, a leukotriene, and then here's LXA4 and LXB4, the lipoxins. All of the lipids mentioned in that passage, both the ones that are high and the ones that are low in the tumor cells, are metabolites of arachidonic acid, and therefore potentially of linoleic acid. They're all different branches of these pathways leading down from arachidonic acid. So you see how ridiculous it is, how simplistic, how cartoonish it is to look at the biology that they report and conclude, oh, so just cut back on linoleic acid, cut back on seed oils. Some of the molecules that are low in the tumor cells and that are anti-inflammatory also come from linoleic acid, also come from arachidonic acid. It just doesn't follow. And in fact, we have dozens of studies showing that when you give people seed oils, you don't see an increase in inflammation. In some, you even see a decrease. We've looked at all of this extensively in previous videos. By the way, I don't put all the blame for this mess on the media and influencer nation, because the authors themselves, in their interviews with the media, hinted at this connection with the seed oils and ultra-processed foods, which is completely unjustified from their findings. I think they were just trying to get attention. They were trying to get a little bit of spotlight. So they mentioned some trigger words, and it obviously worked, but it just confuses the public. Okay, I didn't want to end this video just by explaining this confusion and not giving you guys something more robust to replace that with. I wanted to give you some tools so that you can do this process yourself. You don't need anybody else. The next time around, when there are headlines throwing everybody into a panic, which, let's face it, is probably going to be next week. How can you make sense of this stuff? All these studies being thrown around and everybody on social media saying something different. It's actually surprisingly simple. If I want to figure out the health effect of eating a certain food, food X, there's two basic ways that I can do that. I can look at people eating that food and look at their health, and then compare that to people who don't eat the food or who eat less of the food. This is the most direct way to ask the question. But it's also really hard to do, and it's expensive, and it takes a lot of time because you're following human beings around, and human beings are complicated. So another way to get at the question is to go into the lab and give that food to a lab animal, or even just concentrate one nutrient in that food, isolate it and concentrate it and give it to an animal. Or I can throw that nutrient on some cells in a petri dish in the lab, even human cells, and see what happens. These experiments are much faster, much cheaper, much easier. They're also more indirect. They don't really tell me what happens to a human being eating the actual food, but they can give me some ideas. Hey, I gave these mice vitamin X and the mice got really fat, so maybe a human being that eats a food that contains vitamin X will get fat. Idea. In science, we call these ideas hypotheses. Hey, I threw this amino acid or this protein on some cells and the cells exploded. So maybe human beings who eat a food that contains that amino acid, maybe it's not healthy. Idea. Okay, what do we do with all these ideas? We test them, of course. In science, we always test. How do we test them? We give the actual food to a human being and we watch what happens. Does that sound familiar? It's the direct experiment, right? So the indirect experiments can be useful, can give us some ideas, can narrow things down sometimes. But at the end of the day, the actual result of a human being eating the food is always the bottom line. Such a simple concept, right? You can use this anytime. You can use this every day on social media. You're listening to a podcast and an influencer tells you, hey, lectins are this poison thing. Lectins interact with this receptor or this molecule in the gut wall, and that causes damage to the gut wall, and the lectins cause these other molecules to penetrate into the bloodstream, and that can raise risk of disease A, B, C, and D. Time out. Human beings who eat foods that contain lectins like beans or lentils, are they better off or worse off? Do they have higher or lower risk of disease? Bottom line, hey, dairy causes IGF-1 to go up, and IGF-1 is a growth factor, so that's going to stimulate cell growth. So if you eat dairy, it might raise your risk of developing cancer. Time out. People who consume dairy, milk, or yogurt, or whatever, do they have more or less cancer? Show me that result. Bottom line, such a simple question, and it's always the right question. You don't need scientific training to understand that. It cuts right through all the stories and all the distractions. Okay, quiz time. The study that made the headlines, the Lipidomics study. If I want to know the effect of eating seed oils or linoleic acid for colorectal cancer, is that study direct or indirect? Which of the two worlds? Indirect, right? I didn't look at actual human beings eating a certain food versus eating another. I measured the levels of lipids inside different cells. Generates ideas to test later. Not useless, not bad science. Actually, most of my publications are in this indirect realm, so I'm not biased against it. But we don't jump from these indirect tests to what we should or should not be eating and panic, right? These generate ideas, they generate some knowledge that we then test in the direct type of studies. Okay, this week there was a second study that also came out and also made all the headlines, not as much as the first one, but also got some attention, that also touched on seed oils and cancer. The media was pretty busy this week. So this study was out of UCLA, and what they did, this was a randomized trial. They took participants that had prostate cancer, in this case, and they split them randomly, and half of them received no intervention, so just kept doing what they were doing, no change. The other half were put on a specific diet. They ended up reducing their overall calories compared to the control group, reducing the total amount of fat in their diet, reducing saturated fat, monounsaturated fat, omega-6s, including linoleic acid, and the actual recommendations they received, the participants received, were to cut back on fried foods, highly processed foods, chips, and baked goods. So basically they were told to cut back on junky foods. And they also received a fish oil supplement, so they increased their omega-3 intake, specifically EPA and DHA, these long-chain omega-3s from fish. What the team measured was they got a biopsy of their prostate cancer, and they ran an assay that gave them an index of proliferation, so kind of a readout of how quickly these cells were multiplying, how quickly the tumor was growing. So they got those biopsies and ran that assay before and after 1 year of the diet. And the bottom line is that after the year, the folks on the experimental diet, their index of proliferation was lower. So it suggested that their cancer was growing slower. So really interesting result, right? Quiz time. What type of study is this? Direct or indirect? Which world? Direct, right? Give human beings a specific diet and watch the health effect. See, it's really not that hard. Human beings eating a food or a diet, direct. Mice or cells or biochemical pathway in a petri dish or in a test tube, indirect. So yeah, this type of study is much more in the zone of the type of thing that could lead to dietary recommendations. This was a phase 2 trial. They're probably going to run a phase 3 trial larger and maybe looking at other outcomes. So if this pans out, this diet could be something that gets recommended to patients with prostate cancer. Of course, we can't tell which of the factors that changed is delivering the results. It could be the omega-6s, it could be the omega-3s, it could be the monounsaturated fats or the saturated fats, or the calories or the junk food, or all of it. The trial shows that the overall dietary change seems to have these interesting results compared to the controls. Okay, so what's the effect specifically of linoleic acid and seed oils on prostate cancer? From the large data sets out there that I've seen, people who eat more linoleic acid don't have higher risk of prostate cancer or lower. No significant change either way. And this is from meta-analyses of cohort studies. I've seen a couple of, maybe 2 or 3. A meta-analysis is just when scientists take all the studies on a given question, 5, 10, 15, pool them all together, kind of average them all, and see where is the arrow pointing overall. Is it higher risk, is it lower risk, or no significant change? I don't think these studies settle the issue. It's really difficult to study diet in cancer for many reasons. So I'm open to this going in either direction. The data I've seen so far doesn't support much of an effect of linoleic acid on prostate cancer either way. What about colorectal cancer that the first study touched on, the Lipidomics study? When we look at direct tests of these human beings eating it, what's the effect of linoleic acid on colorectal cancer? What data do we have? I've seen 3 meta-analyses of cohort studies looking at this question. One is from 98. It found no significant association. Another is from 2020. It found that higher linoleic acid intake was associated with a 19% increased risk of colorectal cancer. And the last one is from 2023, and it found no significant association with colorectal cancer. But then when they looked specifically at colon cancer, so just the colon, they found a significant 15% increased risk of colon cancer. So we have some mixed results. Now, just because a study is more recent doesn't mean it's better. An older study isn't necessarily worse. But for meta-analyses, a more recent one generally can include more studies because there's been more studies conducted, more studies published. In fact, that first one included only 4 studies, the second one, 10 studies, and the third one, the more recent one, 15 studies. So I do give more weight to the more recent meta-analyses that show an increase in risk. And I take this seriously, meta-analyses of 10, 15 studies. We don't dismiss that. So what do we do with this in practice? What decisions do we take in our daily lives? One thing we could do is cut back on linoleic acid. We can't eliminate it. It's an essential nutrient. We need to get it from diet, from food. But we could say, okay, let's play safe and get very little, minimize the linoleic acid. And that's a reasonable takeaway from these data sets, from this question in a vacuum. The problem is, when we look at big picture, everything we know about these questions, people who consume higher levels of linoleic acid are also seen to have lower levels of heart disease, lower levels of diabetes, lower levels of total mortality, even all-cause mortality. And all of these are the same level of evidence, meta-analyses of cohort studies. So what do we do with this result with the colorectal cancer that seems to be going kind of against the tide? I'm going to share with you guys my thought process and the work that we did to try to figure this out. But bear in mind, this part of the video going forward is more exploratory. Where do we get linoleic acid in our diet? Well, for most Westerners, it's junky foods. We already talked about it. It's the cookies and the crackers and the pies and the baked goods and the chips and the Burger King and whatever else. So does linoleic acid cause colorectal cancer itself, or is it tracking a low-quality diet, a bad diet, a diet with a lot of junky foods, a diet very low in fiber, which is typical of the Western world, and low-fiber diets are a risk factor for colorectal cancer. So which one is it? Is linoleic acid the problem, or an innocent bystander that's labeling a low-quality diet? We can test for this by adjusting these cohort studies that are used in these meta-analyses for diet quality. Amount of fiber in the diet, amount of certain vitamins, certain factors like that. So I went through all the cohort studies that are included in those meta-analyses. There's 16, total 16 studies. I went through them one by one. It was very interesting. It was a 50-50 split. 8 adjusted for diet quality and 8 did not. So this allows for a really interesting test. If linoleic acid is the cause of cancer, then we should see more cancer with more linoleic acid intake, regardless of adjusting for other foods or not. But if the real problem is the bad quality diets, junky foods, low fiber, and the linoleic acid is just an innocent bystander labeling those diets, then when we adjust for diet quality, when we take that out of the equation, the link between linoleic acid and colorectal cancer should go away. So we ran exactly this test, same process as the meta-analyses do, except instead of lumping all the studies together, we separated them into two groups, those that adjust for diet quality and those that don't, and meta-analyzing them each separately. And for these analyses, I collaborated with Zach Wenger, who is a rock star. He's getting his master's in applied nutrition. And I'll show you the bottom line of what we found, just the gist, the results. But for people who want more detail, I'll link below an article that Zach wrote with all the play-by-play and more analyses and more forest plots. So basically, when we looked only at studies that did not adjust for diet quality, linoleic acid intake was associated with a 28% higher risk of colorectal cancer. And that was statistically significant. And then looking only at the studies that did adjust for diet quality, it was 0%. It was right on the null, so no significant effect. So adjusting for diet quality completely eliminated the effect. And interestingly, when we looked only at the studies that did not adjust for diet quality, the effect was actually accentuated. It was 15 to 19% in the meta-analyses, lumping everything together. And here, 28% looking only at the studies that did not adjust for diet quality. That's the result for colorectal cancer. Looking at studies that report just specifically colon cancer, very similar. When you adjust for diet quality, the effect moves to the null line. So this result is suggesting that what's causing cancer is more the bad quality diet, the junky foods, the low fiber, than the linoleic acid itself. And by the way, we didn't run 10 different analyses and pick the one that looked bad. No, this is the one thing we set out to do, the only thing we did. And actually the results surprised me. I didn't expect it to be this clean. Now, am I completely convinced that this is true? Am I sure of this? Of course not. This is just one analysis that we ran just to get a look at it. It's unpublished, not peer-reviewed. In fact, I recommend you guys take this with a grain of salt. We're going through all the science and trying to figure this out and just sharing this process with you guys. I'm completely open to this being wrong and in the future science showing that linoleic acid causes colorectal cancer after all. Hey, if that's what comes out, okay, we'll go with that. Right now, with the evidence I've seen, personally, my strategy, I don't worry about how much linoleic acid is in my diet or how much seed oil is in my diet. I do avoid junky foods, the cookies and the crackers and the pies and the Burger King. Those are not staples of my diet. So that's my personal approach. Maybe you have a different take and a different personal strategy looking at the same evidence, and that's okay too. For more on seed oils and inflammation, we have a whole video going over that in detail. Check that out right here. And for a lot more on seed oils and heart disease, we got a whole video on that. Check it out right here. I'll meet you in there. Take care.